Good evening, and thank you for joining us in this first of a series of webinars the BNMS is running over the next 6 months on different aspects of research. The live webinars are open access, so if you know of anyone you feel would benefit from attending future webinars, please do pass the relevant information on. The webinars are all being recorded for future reference, and access to the recordings are a benefit for BNMS members through our members page. If you aren't already a member, please have a think about joining our wonderful society. Anyway, on to our webinar. Today's main presentation is entitled Navigating the Regulatory Requirements to Imaging Research and will be presented to you by Professor Gary Cook. Gary needs no introduction to many of us. He currently holds a clinical academic position as Professor of Molecular Imaging at King's College London and Guy's and St Thomas's Hospitals, whilst at the same time acting as nuclear medicine physician at King's and St Thomas's Pet Centre. Gary has a wealth of experience in navigating the sometimes complex twists and turns of the regulatory roadmap. I can certainly attest to finding this somewhat daunting, so I'm very grateful to Gary for sharing his experience and knowledge with us today. To give you a little background, Gary's research interests include imaging bone metastases, measuring tumour heterogeneity, and evaluating novel traces and biomarkers, amongst many other things. Gary's presentation will last roughly 30 to 40 minutes, and for those of you who are able to join us live, there will be the opportunity for you to ask questions at the end of our Q&A session, where Gary will be joined by a panel of experts. The panel members joining Gary this evening will be Neil Patel, Kevin Bradley, and Maggie Cooper. The BNMS Honorary Secretary, Stuart Redman, will be chairing the Q&A session. Now a little bit about our esteemed panel. Neil Patel is a consultant radiologist involved in radiopharmaceutical and oncology research and clinical development at Oxford University Hospitals and in industry. He has an interest in drug development and theranostics and has undertaken both clinical and preclinical research work. Many of you will know Kevin Bradley as the current chair of RSAC, or previously as clinical lead for nuclear medicine at Oxford. Kevin is now Director of Clinical PET Research at PETIC, at the University Hospital of Wales in Cardiff. And Kevin's research interests include PET image optimization, as well as dementia, Parkinsonism, and oncology imaging. And last, by no means least, we have Dr. Maggie Cooper. Maggie, like me, is a radiopharmacist. She's worked in the NHS and in the academia, and I've known Maggie for many years. In fact, she was the radiopharmacist at the Royal Free Hospital immediately before me. And I have a huge amount of respect for her both as a radiopharmacist, but also as my go-to person with difficult research questions, particularly around the regulatory aspects of it. She has carried out translational research in radiopharmacy at King's College London and currently heads up the St Thomas's Pet Centre as their pet chemistry operations manager. I would like to thank Gary and our panel members for giving up their valuable time this evening to share their experience with us all. To keep things simple, we're using a Q&As tab. So if you wish to ask a question, please use this tab to enter it. Please don't use the chat function for questions as they may get missed. You can, however, use the chat function for general comments, information or feedback. At the end of the presentation, Stuart will read out the questions logged in the Q&A section to the panel on your behalf for discussion. So finally, I hope you find this webinar both interesting and useful and look forward to hearing your feedback. I'm afraid I cannot be here for the Q&A session in person. I have an important meeting with my youngest son and a certain Mr. Frank Skinner, tickets for which were bought some time ago as a birthday present. So there we have it. Without further ado, I'll pass you on to the capable hands of Professor Gary Cook. Thank you.
Okay, well, good evening, everyone. Thank you very much for that introduction, Gilly. Just leaves me to also thank the sponsors of this series of lectures, our industrial partners, who we wouldn't be able to put these on without. So the title of tonight's lecture has changed slightly from the beginning. At first, we called it something like the hurdles and barriers to nuclear medicine imaging research, but we thought we should be a bit more positive, and so it's now navigating the regulatory requirements for nuclear medicine imaging research. I don't have any relevant conflicts of interest in giving this talk, but there are a few disclaimers. I mean, what I say is my personal opinion and relates to my experience. I hope everything is factually correct, but the experts in the panel will correct me if I say anything wrong. But obviously things change fairly rapidly in this field, so some things are maybe just slightly out of date. So I would always advise you to consult the Health Research Authority (HRA) website and local R&D departments if you're embarking on a research project. And of course, there'll be institutional differences. So my experience may be slightly different from yours at other institutions. So as Gilly said, I'm Professor of Molecular Imaging here at King's College London and a nuclear medicine physician at the Guy's and St Thomas's PET Centre. We're a university department, but we provide the clinical service for the trust. We're split over 2 sites. So here on the south bank of the Thames, here at St Thomas's, in the Lambeth wing, and in the Cancer Centre over on the Guy's campus. We're very fortunate with our equipment. Maggie heads the radiochemistry department. We have 2 cyclotrons, one for research, one for human radio tracers, and we have a number of scanners. We've just recently installed one of the new Quadra total body PET scanners and have a second on the way early next year. We have a PET MRI scanner also on the St Thomas's campus, and a short axis field of view PET scanner over at the Guy's site. So I've been involved in PET research for as long as the PET Centre has been around, which is 32 years now. And you'd imagine that I've seen some changes over that time. And I say to our clinical fellows, bore them silly by telling them how I could write 2 ethics applications in an afternoon in the old days on a Monday. By Thursday, I would get the approval from the committee, and on Monday we would recruit the first patient. Unfortunately, it's not quite so slick these days, and where it used to perhaps take a week with the wind behind you to get the paperwork done, it's now nearer a year, whether a study is for an investigational medical product, IMP, or not, and we'll come to what that means in a minute for those of you who don't know. But hopefully I'll have a few tips and tricks that will be helpful to those embarking on research and those with more experience. So the first thing to cover is, of course, good clinical practice principles, and I'm more familiar than usual with these because, as you're probably aware, you have to have training on this and update your training every 2 years. Mine just happened to run out last week, so I went through the training course the other day and have updated another 2 years. But there are 13 principles, and I'd recommend that you read these on the Health Research Authority website. But the most important one, of course, is that the right, safety and well-being of the trial participants are the most important consideration. Also of increasing importance in the last 5 to 10 years is the rights, integrity and confidentiality of trial subjects, and those must be protected. And of course, the reporting of clinical trials should give assurance that the knowledge and reported results are credible and accurate. So if you look on the MHRA or HRA website, this is the clinical trials route map that you may well come across. And if your trial does not involve an investigational medical product, and I'll come to the details later, as I said, then you can ignore the circles with the dots in the middle. But everything else for any non-CTIMP or CTIMP trial will have to follow these pathways, and it looks a bit daunting, and now you can understand why it no longer just takes a week to get permission to start a research study on human participants. But when I saw this diagram, which I hadn't come across before one of my PhD students showed it to me actually, it reminded me somewhat of this map, which you'll be more familiar with. So to get from a research idea to recruiting your first subject, i.e. from Amersham to Upminster, you will probably need to change track or lines a number of times. There will be occasional opportunities to run processes in parallel, but unfortunately not always. They're often consecutive, and you'll often find yourself delayed. There'll be cancellations, and often you'll go round in circles, but eventually you can get there to day one of recruitment to start your study. And as I say, I hope I'll be able to give you some information and experience of how to get there this evening. So what are the preliminary considerations? Well, first of all, you've got to decide whether what you want to do is actually research, or whether actually audit or service review might be more applicable, because certainly in our institution, audit projects or service evaluations or reviews are much easier to get the appropriate approvals for internally, and doesn't involve external agencies. And then the most important question overall is whether the trial that you want to run involves an investigational medical product or not. So our tracers are potentially investigational medical products, so IMPs, but not always in a research capacity. So a clinical trial involves an IMP, and this means that the focus of the research is about the radio tracer that we're using. So for example, can FDG diagnose COVID-related pneumonia? So that would be an IMP trial, because your focus is on FDG and whether it works or not. Ways of avoiding having your trial as an IMP or a different angle to the research can be mechanistic trials, whereby you're more interested in the underlying biology and processes rather than the tracer itself. So for example, in that example I just gave you about COVID pneumonia, you may say, does increased glucose metabolism predict outcomes in COVID pneumonia? And you haven't even mentioned the word FDG, because there's a number of ways of measuring glucose metabolism. You know, there's MRI methods, there's FDG PET methods, there's potentially biopsy, autoradiography, and so on. So FDG is no longer the focus of the research. It's looking at the, you know, you're interested in the mechanism. So this would become a non-IMP study. And the reason this is important, because if it's an IMP study, it needs MHRA approval. This, as we'll probably hear from Neil later from the industrial perspective, takes longer. It can be quite expensive. The trial will need auditing, which can be expensive, and it's much harder work. And I would suggest that us state-funded academics, if you like, don't really have the resources to do IMP studies on our own, unless we're in large departments with lots of regulatory support, and that much of our research is probably mechanistic. But when we are involved in IMP research, it's usually on behalf of industrial collaborators who do have more support and experience of getting drugs approved by the MHRA for trials, for example. So the next thing to think about, because as I suggested, this is quite a long-winded process these days, is to check whether there may be an overarching institutional ethics in place for either retrospective or prospective clinical data research. And again, if you're trying out a new tracer for an application, then this won't apply. But if you want to look at previous clinical data or future clinical data, maybe for AI research or radiomics research or something like that, you may find that your institution has an overarching ethics. And so you just have to apply to a subcommittee within the institution, and they will make sure that their ethics application that they've got will cover the work you want to do. And this can take a few weeks rather than several months, but obviously doesn't cover some prospective research of new tracers, for example, that we might be interested in nuclear medicine. So then you need to think about what your research question is, what your hypothesis is, and then the most important document to start with is the protocol. And I found in the past that until you actually sit down and start writing the protocol, that you were never quite clear what your research question was and how feasible or not it would be. So that's a very important preliminary part. And then, of course, you need to know that the tracer you want to use or tracers for your research are available, when they're available, and how much they will cost. And certainly, researchers who come asking for us to help them do PET research, who aren't in the nuclear medicine world, underestimate the complexities of producing tracers for research. And I'm sure Maggie will expand on that either this evening or in her lecture next month. And then, of course, you can't do anything for free these days, so you'll need funding, whether it's from an intra-department slush fund or a charity research or UKRI, or as I say, an industrially supported project. Then it will need to be funded, and you will need to show evidence of that funding. And of course, you need to know that you've got the infrastructure and staff to actually carry out the study in the nuclear medicine or PET department. And also, these days, you can't get permission to do research without having consulted the patients and public. And this can be very useful to making sure that what you thought was a clinically relevant outcome is so, and that the patient information sheet, for example, is in lay language that can be understood by our patients rather than in academic speak. So if you are thinking about doing a clinical trial with an investigational medical product, then you'll need to consult the Medicines and Healthcare Products Regulatory Agency, the MHRA, who oversee the regulation of radiopharmaceuticals and other drugs, of course, in the UK. So all clinical trials with an IMP must be approved by the MHRA, and they will give you a clinical trial authorization, which is required before using an IMP. And you will need to supply them with the clinical trial protocol, the investigator's brochure, and there needs to be evidence of GMP manufacture of the radiopharmaceutical that you'll be using. Every trial on humans requires ethical approval, so you'll need to provide documentation to a research ethics committee who are overseen by the HRA, and they will need documents such as the protocol, how you're going to deal with adverse events, the patient information sheet, the consent form. They'll need to know that you're insured, your institution or the sponsor of the trial is insured, that GDPR is dealt with. And then following the ethics committee approval, then near final approval will come from the HRA. And this is often a process that's in parallel with the ethics application and approval through the IRAS portal, which we'll come to in a minute. Again, just to reiterate that the underlying principle is that of good clinical practice. So this ensures the right safety and well-being of our trial participants and the credibility of the data that you produce. And of course, and we've got Kevin here tonight as chairman of RSAC. This is particularly relevant, of course, to our research, where research approval from RSAC will be required before we administer radioactive substances to research subjects. And this can be applied in parallel through the IRAS portal with the ethics and other approvals that are required. And then we need to think about our protocol design, what the endpoints are. We'll need to get some statistical help to calculate a sample size, for example. And then when we come to the manufacturing of the radiopharmaceutical, I won't go into this in detail because obviously you'll have Maggie to tell us more about this, but radiopharmaceuticals have to be manufactured according to GMP standards, whether it's an IMP trial or not. And that's quite an involved, costly and time-consuming part of any clinical or research procedure. And then the patients obviously need to consent to being part of a trial, and they need to have been informed appropriately of what they're letting themselves in for and any risks that might be involved. So in particular, those related to the radiation doses from our nuclear medicine procedures. And of course, informed consent must be obtained before they start to take part in a trial. Trials, it's highly recommended that non-IMP trials are registered centrally, but certainly CTIMPs have to be registered and require a uterus CT number, and certainly for a non-IMP trial, you have to justify why the trial doesn't need to be registered. And there's a number of places that trials can be registered, but the most common one, and certainly one we've used most commonly, is the clinicaltrials.gov method. And this isn't a 5-minute registration, unfortunately. They need to know quite a lot of detail about your protocol, and often we'll come back with further questions and comments and recommendations about your trial design, in fact. So be prepared. And then even once you've got going with your study, there's ongoing monitoring of the trial necessary, particularly if it's an IMP trial, to ensure compliance with the protocol and regulatory requirements. One needs to be able to report the adverse events should they occur. Any safety concerns obviously must be rapidly reported to your sponsor, which is often the R&D department of your trust, the MHRA and the ethics committee. And of course, data management and reporting very important. It's crucial that the results of trials should be reported in accordance with regulatory requirements, particularly IMP trials. So what's the process? Well, the first step, once you've got your protocol and you've checked that everything is potentially in place, is to gain sponsorship. This is often your NHS trust, but like if you're like me and you work for a university with an honorary position in the trust, then actually we have to get co-sponsorship. So both Guy's and St Thomas's and King's College London end up sponsoring most of our research. But it may be that an industry partner will want to do the research with you and will take on the burden of sponsorship. But this is an application, as I say, usually to your R&D department. And if it is a commercial trial, then a clinical trials office often will deal with the industry partner outside of the central, more clinical R&D department. They'll need to see the protocol. You'll need to complete the IRAS form, which I'll come to in a bit more detail in a minute. They'll want to see the patient information sheet, the consent form. It's recommended that GPs are informed whenever one of their patients takes part in research, and there is a template that we're required to use in our institution. They will need to help you put data sharing agreements together. One of the silly things that we have to do is, because we're a university employee, even though we provide the clinical service and acquire the scans, we have to have a data sharing agreement so that clinical data that's under the governance of the trust can transfer over to the university. So we're in the silly position of having a data sharing agreement with ourselves, but also material transfer agreements and other contracts. For example, we often use an external statistician to help us with our trials, and that requires a contract usually. So all this paperwork needs to be in place, and your sponsor will want to make sure that's all in order. As I mentioned earlier, most trials, well, certainly IMP trials, will need to be registered, and it's highly recommended that non-IMP trials are also, and you need to give a good reason why not. you will need to have consulted the patient and public, and you'll need evidence that you have consulted them and taken their views on board. For us, that can be as easy as just having an email from the manager of our PPI group to say, yes, Professor Cook presented to us, and we fed back. So they don't want fine detail. You will need proof of funding, and this usually takes the part of an award letter, and the dates are important, so they'll need to know that the funding actually covers the planned timing of the study. And then you may want to consider whether to apply for NIHR Clinical Research Network support. And I think this is question 5B on the IRAS form, where it says, do you wish to make an application for this? And the reason for doing this is that if your project is approved by the NIHR, and the various criteria that they use, so if it's been competitively funded, for example, and it is true research, then you can have CRN approval, which means that potentially you can draw on help from research nurses and clinical trial practitioners and so on that your trust or institution may have to facilitate your research. In practice, they're already extremely busy, these people, so you may not get as much of them as you actually need, but in principle, that's approval will allow that to happen. And the advantage for your institution is that they will get additional funding for each patient you recruit to your study, which covers the salaries of those support staff. Your sponsor, so as I say, usually your R&D department will want to see the CVs of certainly the PI and probably the co-Is. They'll want to know that everyone is up to date with their GCP training. And something that I've just recently come across, which has caught us out, is that NIHR recommend that rather than saying the age range for cancer studies should be 18 and above, as we've always done in the past, they feel that younger patients should not be excluded from research, and that we're encouraged to include 16-year-olds and above. The problem with this is that if you're between 16 and 18, then and dealing with these patients of that age, then you need an enhanced DBS, and many of your co-investigators on the study may not have even their DBS up to date, let alone enhanced DBS. So it's just another layer of work that needs to be done before your R&D department will let you go ahead. They will then do a capacity and capability assessment, and this is to make sure that your institution, so commonly your hospital, your trust, has enough capacity to actually help with the research, even if you've got the funding. So for our mainly cancer studies, for example, the head of R&D in the oncology department will need to sign off, the imaging radiology department will need to sign off, and the path labs, if you've got blood and other histology, for example, specimens in your study, they all need to sign off that they're happy that there's capacity to help with your research. And this can take a long time, and you end up chasing people to get their signatures. So this is the IRAS, or Integrated Research Application System, is central to any application, and all applications will need to be done through this system. It's actually quite a good system, which has evolved over the years and is reasonably straightforward considering the complexity of the data that it has to capture. But when you first log in and set up a study, you answer a few questions, and then that opens up the relevant pages for your study. So you can see here, this study, the green bars are the pages that you need to fill in, and the white ones for this particular study, I can't remember which one it was now, but the white ones are those that you can avoid. So for example, this section is about investigational medical products, so we probably had a mechanistic non-IMP study for this. medical devices is blank, so we don't have to do anything there. Ionizing radiation, of course, we do need to fill in, and this is where the RSAC, part of the RSAC application starts. Whether you're taking tissue or blood and samples will determine whether this part of the application opens up, and as you can see, children and so on. So there are many, many pages to fill in, and it can be quite hard work, but everything's reasonably straightforward and well designed, and it's an easy-ish form to fill in online. And when that's completely filled in, you can send a draft to your R&D department, who will then decide on you know, whether they want to offer to sponsor your study or not. So the protocol, what they'll want to see in the protocol is the list of your researchers, co-investigators, including a statistician and study coordinator, if relevant, the background and rationale for your research, the aims, objectives, and hypotheses. And then you may need to argue whether a study is mechanistic versus an IMP study. In the old days, you used to be able to email MHRA and get their advice on whether they thought a study was a CTIMP or non-CTIMP study. And I think that became so popular with investigators, and particularly R&D departments who wanted to see evidence that it was not a CTIMP, for example, that they just became overwhelmed. So I believe it's still now the case that the MHRA recommend that you have an internal committee that will decide on whether a study is mechanistic or not going forwards. And obviously the people on that committee who are making those decisions should not have conflicts of interest with the actual study going on that they're deciding on. Obviously it will include things like the study design, the age range, and mention again about how we're encouraged to include more than 16-year-olds rather than more than 18-year-olds, the inclusion, exclusion criteria, the methods, procedures. And I'd recommend that you don't be too precise in the procedures and methodology of the project. Obviously, you need to make sure that you're able to get the data out that you want and it's reliable, but don't paint yourself into a corner, otherwise you'll end up finding that you want to change things very slightly and you have to go back and put an amendment in. So if you can keep things fairly flexible, which allows a little bit of movement, it will help at later stages. You need to say how you're going to analyze and do the statistics of the data, and there needs to be a section on safety assessment and reporting, whether it's an IMP study or not, and how you're going to handle the data and stay within the GDPR regulation. So this is, as I say, through the IRS system on the portal. You'll also need to persuade either one of your friendly physicists to be the medical physics expert, and a clinical radiology or nuclear medicine colleague to be the clinical radiation expert. They need to be HRA registered. If you don't have one in your institution, then HRA can provide them at a cost, but they will need to give an assessment of the radiation risk and burden to the participants. And then the RSAC application, and what certainly non-nuclear medicine researchers don't realize perhaps is what RSAC actually covers. So the research study itself will need to be approved. The RSAC holder for the study will need to have it on their personal license, the tracer that's being used, but also the employer, the site, the institution will need a license, and this obviously concentrates more on the sort of the infrastructure to produce the safe radiopharmaceuticals. So there's more than one sort of level to this, and Kevin might expand on that later. And I've mentioned the MHRA, which need to be will need to approve a CTIMP study, but not a non-CTIMP study. And the sponsor again, will need to know, or will need to be insured and have indemnity, and the ethics committee will want to see proof of that. So you'll need to show them the insurance documents. So once you've sort of got past your R&D department and sponsorships agreed, then you can apply to an ethics committee. My recommendation would be to choose an ethics committee that you're familiar with, rather than the one that has the next available choice, because some are more familiar with, say, radiation than others, and it may just save you time, even if you've got to wait an extra month to deal with a committee that understands radiation, rather than one that hasn't come across it and comes back with loads of questions. If you can attend in person or online, it'll probably help speed things up, because you can answer questions then and there, and I think it's just polite to do that anyway. So once ethics have approved your study, then it goes back to the R&D department, who then do another capacity and capability assessment to make sure nothing's changed in the several months you've been working on your study setup, and then you'll be given the all clear to go ahead. And then you need to make sure you've got a site file that keeps all the paperwork in one place, a delegation log that names all the people involved in the study and what their jobs are, and it's advisable to sort of set up meetings with all those involved and do some dry runs, particularly if it's a complex study protocol. In the old days, we had to start recruiting our first patient within 70 days of opening, and I think that's been dropped now post COVID, but there is sort of pressure, I think, to get going as soon as you're finally given approval. And unfortunately, the regulatory side doesn't stop once you've started your study, because there needs to be annual reports to the ethics committee, to your R&D department, usually to the funder, and then at the end of the study you need to notify ethics and HRA that the study is finished, and produce a study report within a year of closing the study. So this is the letter that you will hopefully get once you've been approved by the ethics committee, and you think, fantastic, I can get going. But you have to sort of read the small print, and this is a favourable opinion from the ethics committee, but does not allow you to start until the HRA has approved. So that's the next level. And then, so this was another letter from a different study that we got, and then you get the HRA approval, and you think, fantastic, I can go now. And they say, no, hang on a minute, we're happy, but you need to make sure that your NHS organisation has confirmed capacity and capability, as I was mentioning earlier. So, but once you get that, then you're ready to go. And as I say, there's ongoing regulatory requirements. So one of the things that is important, particularly if it's a badged NIHR study, is you need to upload the monthly recruitment figures, even if you haven't recruited any patients during the month, because this goes back centrally, and as I say, funding is attached to the number of patients you recruit under these NIHR approved CRN studies. There'll be research meetings, particularly if it's a CTIMP study, to talk about recruitment, reporting. You may want to feed back to your patients groups, and obviously any adverse events need to be reported. There'll be annual ethics reports, as I said. There may well be some amendments, either minor or substantial. Anything that changes the protocol will unfortunately end up as a substantial amendment, but these, compared to the main approval, are relatively straightforward. But as I said earlier, the more flexibility you can build into your protocol, then the less likely you'll have to put amendments in. And then an end of study report. And you may wish to inform your participants, and there's a bit of the IRS form that actually asks you how you're going to inform participants of the results of the trial. What we tend to do, I mean, working with cancer, you have to be a bit careful that you don't 5 years later send a letter to someone who no longer is with us. So we tend to say that we'll inform participants who contact us and ask us what the results of the study are. I think that's fair, and most ethics committees are happy with that. So to finish off, the basic things we need to first think about is what I want to do actually research? Because if not, then there may be easier ways of doing it. is there an overarching ethics approval in your institution already that you may be able to apply to that will save you a lot of time and effort? And then, as I've been saying, the most important thing probably is to decide whether the radiopharmaceutical you're going to use in your study is a non-IMP or an IMP. Then which tracer, whether it's available, how much it costs, etc. You need to get funding through a grant or some other means, or through industrial support and sponsorship, and then go through the R&D department to get sponsorship, then through the ethics committee, get an HRA approval, back to the R&D department for capacity and capability, and then you're open and ready to recruit, but not forgetting that there are ongoing regulatory reports that will be required. So a bit of a dry subject. Hopefully there's some hints and tips of how to get around some of the more complex aspects of this. And thank you very much for your attention this late on a Tuesday evening in the middle of the week. Thank you.
Thank you, Gary. Look, can you hear me, Gary?
I can, yep.
Great.
Sharing.
Thank you for your excellent talk. I feel reassured that there's enough governance around this, which is great news. I also feel slightly amazed that any research actually gets over the line. Very impressive. Excellent. So there is a question in the chat, which I think I might, I'll get to in a minute. I may just ask some of our august panel just to dive into the areas of their expertise a little bit, and I'm going to bring that question in during that. So if I could start with you, Kevin, and just to remind everyone in the audience, Kevin's obviously a consultant in Cardiff, but also the chair of the Administration of Radioactive Substances Advisory Committee. So from an RSAC perspective, what is the responsibility of RSAC in this area? When
well, it's got a legal responsibility in that any administration of a radioactive substance to a human throughout the UK requires an RSAC license. And as Gary quite rightly stated, that is for the practitioner, i.e. the responsible clinician, and also for the institution, or it's called the employer under radiation regulations. So one needs two RSACs, or two green lights, if you like. For people out there who may not be aware, that RSAC is composed of multidisciplinary nature. There's about 25 members usually at any one time, and most of us are actually involved in research and clinical practice. So we want to facilitate research, and I think we do. And sometimes the submission to RSAC can be, shall we say, a little bit lacking or a little bit contradictory, and we often just tidy things up for people. So we very much facilitate research. And probably about 500 applications a year, circa. I hope that helps.
Yeah, and as a follow-up question then, is there anything that people can do, any obvious things, I know this is generic, to smooth the kind of process through RSAC? So simple things that fall down. I know when I was on RSAC with you, there was, some things flagged up that you thought people could.
Well, I think someone sensible and senior needs to perhaps reread what's been submitted, because I think the one thing about IRAS is that maybe there can be multiple edits by different people, and that can lead to conflicts. And one does want to see consistency. So if the protocol says that there will be 10 scans in total, and then part of the IRAS elsewhere says there are going to be 3 monthly scans for the next 5 years, then there's a conflict and it's going to lead to questions. It all gets tidied up, but that leads to a delay.
Yeah, the to and fro is difficult. Yeah.
In general, I would say that most UK research submitted by the likes of Gary, for instance, or other people who are on this call, pretty well goes through in a straightforward manner. I think that increasingly, actually, some of the delays have been with foreign-sponsored research from drug companies who use a CRO in the UK, and there's, as I say, conflicts between the protocol, between IRAS, and between what's been requested. And at that point, one has to be certain that it's going to be a safe practice and how much radiation that the patient or volunteer is going to be exposed to.
Yes.
It is, RSAC is on people's side, but it's on the side of the patient first and foremost, and then it's on the researcher's side, and it just tries to keep everything safe, simple, and transparent.
Yeah, I think that's reasonable, and I found the office very good at RSAC and organised at what they do. And on the other side of it, as a clinical radiation expert, the HRA are very good. They really know what they're talking about, and they're good at tidying up and trying to work with applicants to tidy up those applications so that all those things line up. So I think there's a genuine desire from RSAC and from HRA to help get through this process rather than try and put blockers in.
Oh, no, absolutely.
Whether it feels that way on the other side, I'm sure sometimes it doesn't, because you just want it to go through. But yeah, there we are. Great. So coming to Maggie, so from a kind of radiopharmacy side of things, are there any tips for navigating the radiopharmacy regulatory requirements? I know you might get further into this next month, but
well, I mean, the main thing that, like Gary said, is to work out whether it's an IMP or not an IMP. Because if it is an IMP, then you're going to need a QP to release that, and that's going to be a big challenge for you if you don't have that already on site. But the main thing also regarding the IMP is that if you don't have a license to manufacture IMPs in your site, then that's also a massive hurdle to get over. Now, the government are changing the legislation so that diagnostic IMPs can be manufactured without an IMP license in your radiopharmacy, but it's not completely clear from the MHRA exactly how they're going to roll that out. So for the time being, if you're doing an IMP study, you need a QP, you need an IMP license, and that's very, very challenging, I would say. If you're doing therapy, you're always going to need it's almost certainly going to be an IMP, isn't it, if it's therapy anyway? So then you're going to need a QP and all that goes with that, with a license. Yeah.
Yeah. Excellent. Fine. Good. So Neil, I won't come to you just for a second, or I might come to you. I'm going to take some of these questions, if that's all right. What are the additional considerations, if any, for using healthy volunteers in new IMP studies? So I don't know if Neil or Gary wants to take that or start with it.
Yeah, I've not been involved in any. It might even be Kevin is best placed to answer this because I know RSAC have a view on healthy volunteers, and there's different levels of accepted radiation. But in this era of total body PET now, where we can get the injected doses right down, I think it's going to become a more common aspect of nuclear medicine research.
Yeah, Kevin?
I agree with that. There is an expectation with healthy volunteers, particularly if they are young volunteers, that their total burden of radiation should be kept to an absolute minimum. And there is guidance out there, and that level of radiation, I mean, I could quote a figure, ballpark figure, because everything is individually assessed, of 10 millisieverts, should not be exceeded if at all possible, but it is on a case-by-case basis. Gary alludes to total body PET. Indeed, most of the normal volunteer studies are probably microdosing studies that actually don't even involve imaging, but involve samples being taken. But for instance, other investigations, new devices, because devices can be subject to MHRA review as well. But for instance, devices, if someone changes an inhaler or a nebulizer, they may use radioactivity as part of that, and that will be potentially in normal volunteers to start with as phase 1, and then go on to patients, relevant patients.
Yeah.
So it is just common sense and safety. We do not want to inflict unnecessary radiation to completely normal volunteers who pretty well by definition will not benefit directly from that radiation. And the rules change depending on risk.
Okay, great, thanks. So there's the next question is about the 16 to 18-year-old group, which there's obviously a shifting kind of view amongst the kind of regulators and the drivers in this area. I was going to ask Gary, how much harder is the getting something through for the pediatric group? First of all, in that 16 to 18 group, is it, if you were starting on the front of a study to include those, do you think you would need a lot of extra time and effort to get that through?
No, I mean, I think the only practical difference is to get that enhanced DBS or your study team to include the 16 and 17-year-olds. I think, you know, realistically in oncology studies, most of the stuff we do is in, you know, over 50-year-olds. So it's not actually relevant, but obviously it may be. I've no experience in doing pure pediatric research to be able to sort of comment. But certainly if I was asked to be a PI on a pediatric imaging study, I would go and seek help before even attempting to do it myself, that's for sure.
And getting into like the MRT space and things, it's less common, I suppose, but apart from maybe neuroblastoma and things. I don't know. Have you got any experience, Neil, of pediatric applications?
No, not just that. We try to avoid it as much as possible.
Just because you know it's harder.
Yeah.
Okay, fine. So the next question is probably coming your way, Maggie, because it talks about homebrew radiopharmaceuticals, which is making me anxious. But can you see that question there?
Yeah, I see the question. I also laughed because I was like, really, you still have to make it to GMP standards, and you still have to go through all the things that I'm going to talk about next week, which is eye-wateringly painful. And I think that it will be very informative for everybody to see what we have to do on the radiochemistry side of things to ensure that everything is completely compliant. So I also would throw that to Kevin, because you know, if you're going to make something that's kind of you know, isn't really going to be tested properly, or maybe hasn't been in humans before, I think that our site would be quite interested to know, you know, what tests you've done. What, you know, you can't just have an idea and just bung it into patients. You've got to have some idea of if it's safe and that type of thing. But I mean, there are things that you can do. I mean, we've done ourselves. I mean, Gary can tell you there are stuff that we've done that's gone through the research lab into the GMP lab and out into the clinic, and it's fine for some sort of mechanistic studies without actually going through a full clinical trial as such, because it was a mechanistic study. But I still think that you can't. There are no shortcuts, I would say. Unfortunately, it's just what if it's going to come through radiochemistry, it's going to have to, or radiopharmacy, it's going to have to be done to that same standard, regardless of whether it's a kind of homebrew or a full trial.
Okay.
Could I?
So I'm not going to go to Kevin, because I'm going to make sure I get Thomas Wagner's questions in, because he's asked two, and I'm not ignoring you, Thomas. So I'm going to first of all, I'm going to take a kind of industry perspective on this. So what is a reasonable timeline from idea to first patient recruited? I'm going to come to you, Neil.
I think that depends on how much resource you throw at it. And also geography. So we've just finished a trial where it was 3 months from conception to completion of the trial and getting a clinical study report. That's one end of the spectrum where
that's aspirational.
Wow.
Yeah,
we threw the bucket at it.
you wouldn't like everyone to leave thinking that's normal.
Yeah, there are set timelines that you can't avoid, such as the MHRA turnaround times of like 60 days, etc. Which so and then there's the other end of the string, the spectrum, where I've had a trial where we've taken over 2 years to get it up and running. And that's from a variety of different issues, not really from the regulatory side of things, but more actually from internal. I think one of the biggest issues I've experienced as a researcher are some of the institutional or local issues of trying to get this trial set up.
Yeah.
And just simple logistics of getting patients to a department, etc., have actually been things that hold it up more than actually the regulatory side of things.
Okay. So we've only got a minute or two left, so I'll just try and get through a last couple. Does FDA approval for radiotracer for diagnostic PET exclude the requirement for CT MRHA approval? I'm guessing not, but Maggie? Must help. Oh, mute. Fight the mute button.
I don't know. It's still got to go through. I mean, if you've got that data, it helps, doesn't it, with the application to the MHRA, but it doesn't exclude the fact that you have to do the same application. So, I mean, we've had things where it's been made in another country and we've still then have to go through all the same tests and stuff, but it's a little bit simpler, but it's not, doesn't mean you can't, it's not just, it's not easy. It's still the same process.
And there's a follow-up question about the category of the study, how best to get into that.
There's a question after, which is about low-dose lutetium. I mean, if it's purely being used for diagnostic studies, then in the future, I think we'll be able to get these through without having to go through having an IMP license for the manufacturer and without having to have a QP release. That's fine, but I think that you can't then be using it for therapy if it's it really has to be for diagnostic use, and I think that will be scrutinized. So I don't think I just don't think you can bend the rules, unfortunately.
So I think we're coming to an end now. So I'd like to thank Gary again for his excellent talk. It was perfect. I think it covered all the ground that we required. I'd like to thank the panel members for your expert opinion. It's very kind. I'd like to thank the sponsors, and they're in the chat as well, Curium, SciTech, and Siemens Health and Ears. It's really grateful for you coming on board with this. It definitely helps. I'd just like to mention that we take this forward another step next month with Maggie, who'll be presenting on the radiopharmacy side of things. So there's someone just following up back in the chat. And lastly, I'd like to thank everyone for attending. This is great. It would be great for people to get as involved as possible, because clearly we need the answers that the research is going to generate. So thank you all.
Thanks, Stuart. Bye-bye.
Okay.
Bye.
Thanks, Stuart. Thanks, Gary. Cheers.